Good day all. I welcome you all to today's edition of Dr. Ife's Hematology and Blood Transfusion Science Lecture Series. Today we're going to be discussing Kell blood group system. At the end of this lecture, it is expected that you should be able to mention the antigens that are in Kell blood group system, also the antibodies and their reactivity. You should also be able to explain the genetics and inheritance of Kell antigens, and of course explain the clinical significance of Kell antibodies. Introduction. Kell blood group system is a complex system. It is polymorphic. It has many phenotypes. And the antigens that are present in Kell blood group system are also immunogenic, but they are third potent antigens only after ABO and RH blood group system are triggering immune response. They are only after ABO and RH blood group system, so they can say that they are the third potent antigen and triggering immune response. History. The Kell blood group system was discovered in 1946, and it was named after Mrs. Keller, a patient in whom anti-Kell antibodies had resulted in hemolytic disease of the newborn, of her newborn child. Anti-Kell antibodies resulted in hemolytic disease of the newborn of her child. The Kell antigens are expressed in, they are geographically distributed. They are expressed in different frequencies in different populations. The antigens that are found in Kell blood group system, we have up to 35 antigens present in Kell blood group system. And the ISBT number for Kell blood group system is 006. And some of these antigens are named K1, Kell1, Kell2, Kell3, and so up to 35. But also have an alternate name, like Kell1 is designated as big K, while Kell2 is small k. Then you have KPA, that is called Kell3, and then KPB, that is called Kell4, and then JSA, Kell6, JSB, Kell7, and all the rest of it as shown on the slide. But the two commonly encountered antigens in Kell blood group system is big K and little k. The big K and the little k antigen, that is the big K antigen and the little k antigen, are the most encountered antigen in this blood group system. Remember, the big K can also be called Kell1, the little k can also be called Kell2. But the big K antigen is the most clinically significant of all the Kell antigens. The DK antigen is the most clinically significant of all the Kell antigens. Just as we discussed when we were treating RH blood group system, the Kell antigens are proteins, just like antigens of RH blood group system. The Kell antigens are proteins. And these proteins, the polypeptide that made up these proteins have about 732 amino acids. The polypeptide chain that made up this protein, the Kell antigen, has about 732 amino acids, and they are glycosylated at about 5 different sites. 5 different sites on the polypeptide chain. This Kell glycoprotein is a transmembrane single-pass protein that carries the Kell antigen. The glycoprotein is a transmembrane single-pass protein that it traverses through the red blood cell once. The single-pass protein that passes through the red blood cell once, and it carries the Kell antigen. It carries the Kell antigen. And the sequence of the amino acid determines the specificity of the most of the Kell antigens. The amino acid sequence determines the specificity of the Kell antigens. The Kell protein, that is the Kell antigen, is anchored to the surface of red blood cell by being linked to an integral red blood cell membrane protein called SK protein. The Kell protein, the Kell antigen, is anchored on the surface of the red blood cell by being linked through a disulfide bond to an integral red blood cell membrane protein. And the name of this protein that anchors the Kell antigen to the surface of red blood cell is SK protein. SK protein. And this SK protein is a transmembrane protein that traverses through the red blood cell membrane 10 times. It traverses through it's a multi-pass protein that traverses through the red blood cell membrane 3 times. And SK protein, if it is absent in an individual, can give rise to what we call a multisystemic syndrome called MCLEOD syndrome. MCLEOD syndrome. Absence of SK protein gives rise to a multisystemic syndrome called MCLEOD syndrome. Remember I said that Kell antigens are proteins. They are made up of polypeptide chain that are glycosylated at 5 different points. And this glycoprotein that carries the Kell antigen is a single pass protein that traverses through the red blood cell once. Now this glycoprotein that carries the Kell antigen are actually anchored to the surface of the red blood cell by being linked through a disulfide bond to a red blood cell membrane protein called SK protein. And this SK protein is a multi-pass protein that has traversed through the red blood cell membrane into 10 times. And absence of this SK protein in the cell membrane gives rise to what we call MCLEOD syndrome, and is a multisystemic syndrome. We'll look at that more as we go on. Genetics of Kell antigen. The Kell genes encodes the Kell antigens. The Kell genes encodes the Kell antigens. And This Kell gene is found on chromosome 7. The Kell gene encodes the Kell antigens, and it is found on chromosome 7. And like I said earlier, I said this gene is highly polymorphic. It is highly polymorphic. And the two major antigens, the big K and the little k, the only difference between them is just amino acid, one amino acid difference. There's a single nucleotide single nucleotide polymorphism chain that changes an amino acid at position 193, thrown at position 193. So in BK antigen, so methionine in position 193 in BK antigen. I repeat that again. The change is just a single amino acid change that occurred at position 193 in little k antigen, changing threonine to methionine, from threonine in little k antigen to methionine in BK antigen at position 193. The gene is located on chromosome 7, and the gene that codes for production of Kell antigens is called the Kell gene. Frequency of Kell antigen. Kell antigens such as little k, KPA, KPB, JSB, Kell 11, 12, 13, 14, 18, 19, and as shown on the slide, is found approximately 100% in most of the populations. You can see it almost 100% in most of the population, be it Black, Caucasian, Asian, and all the rest. Why the BK antigen is rarely found? Like you have only 2% of it in Blacks, the BK antigen, and you have 9% in Caucasian, and you have up to 25% in Arabs. And then you have the JSA, about 0.01% in Caucasian, 20% in Blacks. And then you have others that are also very rare, as shown on the slide. Expression of Kell antigens. The Kell antigens can be found on blood cells of erythroid origin, that is, red blood cells and their precursors. They can also be found in myeloid tissues. The Kell antigen is also expressed in a small amount on a number of organs, including lymphoid organs, muscle, both cardiac and skeletal muscles, and then the nervous system. Remember, note this point that Kell antigens can be found on erythroid blood cells of erythroid origin, that is, red blood cells, both the mature and the immature ones, the red cells and their precursors. They can also be found in myeloid tissues and also in lymphoid organs, both muscles like cardiac and skeletal muscles, and then the nervous system. Then functions of this Kell glycoprotein. Remember, I said that Kell glycoprotein is a single pass protein that traverses through the red blood cell once, and is actually what is actually carrying the Kell protein. And this glycoprotein is anchored on the surface of red blood cell by being linked to SK protein, which is a multi-pass protein, through a disulfide bond. It is SK protein, which is a transmembrane protein that is anchoring the Kell protein onto the surface of the red blood cell. Now, what is the function of this Kell glycoprotein? The Kell glycoprotein has an enzymatic function. It has an enzymatic function. It is an endothelin 3 converting enzyme. It is an endothelin 3 converting enzyme. It cleaves the inactive precursor big endothelin 3 and changes it to active form of endothelin 3. And this active form of endothelin 3 is a vasoconstrictor. It is a vasoconstrictor. So the main function of glycoprotein is the conversion of the inactive form of big endothelin 3 to active form of endothelin 3. And this endothelin 3 is a vasoconstrictor. So it is an endothelin 3 converting enzyme. The Kell glycoprotein is endothelin 3 converting enzyme. Antibodies that are produced against Kell antigens. Just like we said when we were discussing Rh blood group system. In Kell blood group system, the antibodies are immune. They are not natural occurring. And what that means is that they are formed in response to immunization. They are formed in response to immunization. And they are mainly IgG antibodies. You hardly see IgM. They are mainly IgG antibodies. And because they are IgG antibodies, you can also say that they are warm antibodies. They react optimally at 37 degrees Celsius. They can activate complement, and they can cause severe hemolytic transfusion reaction. They can cause severe hemolytic transfusion reaction like anti-BK, anti-little k, anti-KU. They have been implicated in causing, severe transfusion reaction. They can also cause hemolytic disease of the newborn and also severe anemia. They can cause hemolytic disease of the newborn, and they can also cause severe fatal fetal anemia in the newborn. Anti-K, anti-BK, and anti-little k, like I said earlier, they are usually immune. They are immune antibodies. Normally, they are IgG and can activate complement. They usually react, best at AHG, anti-human globulin state, and the use of enzyme technique at 37 degrees Celsius. And some examples may react at 20 degrees Celsius, but just very few of it. Some of it also, especially anti-BK, react poorly in low ionic strength saline. Some of the anti-BK react poorly in low ionic strength saline. They are both clinically significant, capable of causing hemolytic disease of the newborn and hemolytic transfusion reaction. Both anti-BK and anti-little k can cause hemolytic disease of the newborn and also hemolytic transfusion reaction. It can be fatal or severe. Common Kell phenotypes. Like I said earlier, the Kell blood group system is complex, and the Kell locus is highly polymorphic and gives rise to many Kell antigens. There are, however, two major codominant alleles that produce two important antigens, the big K and the little k. And I said that they only differ by a single amino acid at position 193. The little k antigen is more common. The little k antigen is more common, like we said, like I said when we were looking at the frequency. The little k antigen is more common than the big K antigen. You have the little k antigen being present in almost 98% blacks and then 91% Caucasians. Little k antigen being present in almost 98% blacks and then 91% Caucasians. While you have big K antigen being present, just 2% blacks and then 9% Caucasian. And then phenotype, K minus, big K minus and big K plus, small K plus, as shown on the slide. The phenotype big K minus and small K plus phenotype is found in 98% blacks and then 91% Caucasian. Another phenotype that we want to look at is what we call null phenotype. Null phenotype. The Kell system has a rare form of phenotype where an individual red blood cell lacks all the Kell antigens. That is K0, called K null. The individual with this phenotype are healthy, but they produce anti-KU, which they encounter when they encounter red blood cells that do not have Kell antigens. The individuals that have this phenotype, null phenotype, with regards to K antigens, that is, they don't have any K antigen on the surface of the red blood cell. These individuals, they are apparently healthy. But what happens is that if they are in need of blood transfusion and they are transfused with blood that contains any of the Kell antigens, they can produce what we call anti-KU. This anti-KU can react, when produced, can cause blood transfusion reaction because it can react with blood from any other person that has any form of Kell antigen. So for this particular set of individuals, when they are in need of blood, what they should be transfused with is just the individual that will donate blood to them is this individual with the same null phenotype. They can K null phenotype. They can only collect blood from people that have Kell null phenotype. Otherwise, they will produce this antibody, which is anti-KU, that can react with any blood that has any Kell antigen. Therefore, if K null individuals ever require a blood transfusion, they should only be transfused with K null blood products. Okay? There's MCLEOD syndrome. Like I said earlier, that in the red blood cell, the Kell glycoprotein is covalently linked to the SK protein. This SK protein is a transmembrane protein that transverses through, is a multi-pass protein, and a transmembrane protein that transverses through the red blood cell 10 good times. And one of the functions of this SK protein is to anchor the Kell antigen onto the surface of the red blood cell. But found out that in some individuals, they don't have this SK protein. They are not able to produce this SK protein. These individuals are people that develop this multi-systemic syndrome called MCLEOD syndrome. MCLEOD syndrome. In the absence of SK, a condition called MCLEOD syndrome develops, and K antigens are weakly expressed, and the red blood cells are abnormal with spiky projections. In these people that have this type of syndrome, you have a lot of acanthocytes, what we call acanthocytosis, being present. And also systemic findings that you can also see in individuals with this type of this MCLEOD syndrome. You can have muscular dystrophy, cardiomyopathy, psychiatric disturbances, neurological defects such as loss of reflexes and movement disorder. Remember, MCLEOD syndrome developed as a result of absence of SK protein. SK protein is a protein that anchors the Kell antigens onto the surface of the red blood cell. They are covalently linked through a disulfide bond to the glycoprotein, Kell glycoprotein. protein. And absence of this escape protein gives rise to a multisystemic syndrome called MCLEOD syndrome. Clinical significance of Kell antibodies. Okay, one of the most important clinical significance of Kell antibodies is they are capable. They can cause hemolytic transfusion reaction. They can cause hemolytic transfusion reaction, and this happens when a blood that contains a particular Kell antigen is transfused into an individual that lacks that particular antigen. An antibody, a specific antibody, can be produced. Remember, we said these antibodies are immune. So if an individual having little k antigen is transfused into blood of individual with little k antigen is transfused into a recipient that lacks little k antigen, he can produce little k antibody. And if subsequently the same blood is transfused again, this individual can react adversely to that particular blood. It can be fatal, it can be severe. Okay? The antibodies that can capable of causing hemolytic transfusion reaction that have been implicated in hemolytic transfusion reaction is anti-BK, anti-little k, anti-KPA, anti-JSB. And then the production of anti-KU is also common in patients who are with the phenotype null K null. This individual, when transfused with any blood that contains any Kell antigens, they are capable of producing what we call anti-KU. And when this antibody is produced, this individual can react to blood of any individual that contains any Kell antigen. So to avoid sensitization of this type of individual, they must be transfused with Kell null blood products. Kell null blood products to avoid sensitizing them. Another important clinical significance of Kell antibodies is that they are capable of causing hemolytic disease of the newborn. And also they are capable of causing severe anemia, severe fetal anemia. Why? When we are discussing RH and ABO, remember we said ABO can also cause hemolytic disease of the liver, ABO antibodies, but it's not as severe as that of RH antibodies. We explained all that when we had that lecture. But in this case, the hemolytic disease of the newborn, remember I said that K antigens can be found on the mature red cells. They can also be found on red cell precursors. That is the problem. They can be found on the surface of the mature red cells. They can also be found on the surfaces of red cell precursors. So what happens is that these antibodies, when it is produced, goes into baby circulation and destroy the red cell precursors. They destroy red cell precursors, and in doing so, they suppress erythropoiesis. They suppress erythropoietic activity, number one. Number two, these red cell precursors, because they are not well hemoglobinized, they are not well hemoglobinized. So the bilirubin that is formed as a result of destruction of red blood cells are not much. Unlike in HGN caused by RH, the mature red cells are destroyed more than the precursor. So you have a lot of bilirubin being formed, and the baby is born with severe jaundice. But in this case, the precursors of red cells are being destroyed. The precursors of red cells are being destroyed, but there is because they are not well hemoglobinized, you don't have much bilirubin being produced. So the level of jaundice is not as severe as that of the one seen in RH, the HGN due to RH blood group system. But a baby is born with severe anemia that can be severe anemia that can be fatal if there is no form of intervention. So what am I trying to say? What I'm trying to say is that antibodies of Kell blood group system, especially anti-BK, can cause hemolytic disease of the newborn. In addition to causing hemolytic disease of the newborn, they can also induce severe anemia in fetus because they can suppress erythropoietic activity because they destroy red cell precursors, in addition to also destroying the mature red blood cells. So a baby is born having hemolytic disease of the newborn with less bilirubin being formed than the one found in HDN due to RH blood group system. Okay, like I said here, unlike RH and ABO, Kell antigens are expressed on the surface of the red blood cell precursors, and anti-K promotes the immune destruction of K plus erythroid early progenitor cells by macrophages in the fetal liver, rather than only mature fetal red blood cells. Because the red blood cell precursor do not contain hemoglobin, less bilirubin is released during the hemolysis. The jaundice in the newborn period is less common. However, the underlying anemia may be severe. There are antibodies that have been implicated in this hemolytic disease of the newborn: anti-BK, anti-L2K, anti-KPA, anti-JSA and JSB, and anti-UIA. These antibodies, but the chief among them is anti-BK, capable of, because remember we said they are immune antibodies, they are IgG antibodies, they are capable of crossing placenta once formed to harm the unborn child. But this happens in women who have had multiple pregnancies, or several blood transfusions, women who have had multiple pregnancies or several blood transfusions. Once these antibodies against K antigens are formed, these antibodies, because they are immune antibodies, they can navigate through the placental barriers and harm the unborn child by destroying the red cell precursors in addition to the mature red blood cells. And this will induce severe anemia in the baby and also cause hemolytic disease of the newborn. But the jaundice that is formed as a result of this is less severe when compared to that found in RH blood group system. Okay. Thank you. This is the end of today's class. I just hope that you've learned something. I think at this stage you should be able to give an overview what the Kell blood group system is all about, the antigens in the Kell blood system. And I forgot to mention that the inheritance of Kell blood group system is by Mendelian fashion. That means whatever it is that is found in the parents of the mother and the father will also be seen in the child. Thank you very much. Feel free to leave your comments. Thank you.